The cell cycle. G1 phase, the period prior to the synthesis of DNA. In this phase, the cell increases in mass in preparation for cell division. Note that the G in G1 represents gap, and the 1 represents first, so the G1 phase is first gap phase. S phase, the period during which DNA is synthesized. In most cells, there is a narrow window of time during which DNA is synthesized. Note that the S represents synthesis. G2 phase, the period after DNA synthesis has occurred, but prior to the start of prophase. The cell synthesizes proteins and continues to increase in size. Prophase. In prophase, the chromatin condenses into discrete chromosomes. The nuclear envelope breaks down and spindles form at opposite poles of the cell. Metaphase. In metaphase, the chromosomes are aligned at the metaphase plate, a plane that is equally distant from the two spindle poles. The nuclear membrane disappears completely. Anaphase. In anaphase, the paired chromosomes separate and begin moving to opposite ends of the cell. Spindle fibers not connected to the chromatids lengthen and elongate the cell. At the end of anaphase, each pole contains a complete compilation of chromosomes. Telophase. In this last stage, the chromosomes are cordoned off in distinct new nuclei in their merging donor cells. After these changes, mitosis is largely complete, and the genetic contents of one cell have been divided equally into two. Cytokinesis. Cytokinesis is the division of original cell cytoplasm, beginning prior to the end of mitosis and complete shortly after telophase. At the end of cytokinesis, there are two distinct daughter cells. Cancer is a cell division disorder that results in uncontrolled cell growth and is caused by mutations to the proto-oncogene gene and the tumor suppressor gene. During G1, when the conditions are favorable, certain proteins stimulate the cell to begin copying the DNA. Likewise, if the cell is not healthy or large enough, or the environmental conditions are not favorable, the cell cycle stops. The cell cycle can also cease during G2 phase at the DNA replication site. If the DNA is determined to be without blemish, the process continues. If damaged, the cell is suspended until the DNA can be repaired. If a mutation occurs in one of the genes that controls or regulates cell growth in any number of ways, the corresponding protein may not function correctly, allowing the cell to proceed without interruption.